And that's great. We can see it. Don't forget to unmute yourself. Yep, you've done that too. Yes, can hear you, can see it. All good.
Perfect. Okay, so hi everyone, and thank you for joining this session. My name is Karina Santiago Gonzalez, and as Tom mentioned, I am a PhD student at Newcastle University. I'm working as part of the Chicken Stress Project under the supervision of Tom Smulders. And today I will be talking about the role of the avian hippocampal formation in stress regulation. So just as a quick overview of my presentation, I will be talking about the origin and evolution of the hippocampus, the function of the hippocampus, what is stress in birds, and the hippocampal regulation of the stress in birds. So let's start. With a long and ancient evolutionary history, the hippocampus is generally considered a highly conserved region present in most vertebrates. As the brain develops, the hippocampus originates from the medial edge of the telencephalic pallium, also known as the medial pallium. So while in most amniotes, it typically lies in a dorsomedial location, in mammals and in ray fin fishes, it has been displaced, occupying posterior and dorsolateral positions. So comparative neuroanatomy studies have revealed many similarities between the hippocampal formation across different taxonomic groups. In fact, evidence from molecular and genetic studies has shown that many of the genes involved in the development and function of the hippocampus are highly conserved across species. So thanks to this evidence, we can formulate some speculations on the relationships between the anatomical components of this structure across different groups. So let's take a look at the cross-section of the mammalian hippocampal formation. So the mammalian hippocampal formation has a distinctive structure of two interlocking Cs. And it has a trilaminar organization and consists of several subdivisions, including the dentate gyrus, the subiculum, and the cornu ammonis. Each of these subdivisions is unique. This means that they have their own histological, neurochemical, and connectivity properties. Meanwhile, the avian hippocampal formation, here in a coronal section, is less well-defined. And it consists of densely packed heterogeneous populations of neurons. Some authors proposed three main subdivisions: the ventral one, the dorsomedial, and the dorsolateral. And while other more detailed subdivisions, like the one here, have been proposed, there is no real consensus about the number and the boundaries of these subdivisions. However, despite the differences, there are some striking similarities between this structure across different groups. For example, based on connectional and expression patterns of regulatory genes seen during early development, the V-shaped layer of the avian hippocampal formation has been proposed to be an homologous of the mammalian dentate gyrus, while the dorsomedial and the dorsolateral subdivisions appear to show some similarities to the CA1 and the subiculum of the mammalian hippocampus. Yet, as different evidence can suggest different relationships, the controversy remains over the proposed homologies. And plus, let's not forget that the connections between the hippocampus and the rest of the brain changed considerably during evolution with the emergence of the neocortex, adding an extra layer of complexity to this whole situation. Despite the challenges, the hippocampus remains an intriguing and essential subject of study. Its remarkable conservation across vertebrates is a clear indication of the great evolutionary and functional significance of this structure. So let's talk about the function of the hippocampus. So the hippocampus performs several functions that are critical for survival and adaptation. I'm sure that for most of us, when we think about the role of the hippocampus, the first thing that comes to our minds is its role in spatial navigation and memory. We all rely on the hippocampus for navigation. Birds use the hippocampus as part of their migration and to navigate their environment and retrieve stored food items when food is scarce. But what most people may not realize is that the hippocampus is also crucial for regulating our emotions, including the way we respond to stress. The current thinking is that these functions of the hippocampus are separated across the longitudinal axis of the hippocampus. So this means that in mammals, the dorsal portion of the hippocampus appears to be more involved in processing information related to spatial navigation and episodic memory, while the ventral portion is more involved in processing information related to social and emotional contexts, including stress regulation. But what about birds? So in our lab, we're working to answer this question and investigate whether the caudal portion of the hippocampus, the potential equivalent of the ventral mammalian portion, is involved in regulating the stress response. Although we have some evidence to suggest that the avian hippocampal formation is implicated in the stress response in birds, the exact mechanisms and the neuronal pathways involved are not yet well understood. So how do birds actually cope with stress? The stress response mechanisms appear to be evolutionarily conserved across vertebrates. This means that, just like us, birds respond to stress by the activation of the hypothalamic pituitary adrenal axis, which is one of the key systems involved in the stress response in vertebrates. So neurons in the paraventricular nucleus of the hypothalamus start a cascade of events involving the release of corticotropin-releasing factor and arginine vasotocin. Together, they stimulate the release of adrenocorticotropic hormone in the anterior pituitary, which in turn stimulates the adrenal cortex, resulting in the release of hormones such as corticosterone, which is the major glucocorticoid in birds. This cascade of events puts the system into action. And once the system is active, how do we turn it off? So determination of the stress response is crucial. We know that chronic or excessive activation of the stress response can lead to negative health outcomes, including, for example, anxiety and depression in humans, and also to poorer welfare in other animals. So this termination is mediated by a negative feedback system in which glucocorticoids bind to receptors at different levels within the axis, including the hippocampus. So how do we know that the hippocampus turns off this stress response? Number one, the hippocampus of both mammals and birds contains the highest concentration of glucocorticoid receptor sites, allowing it to play a critical role in the inhibitory feedback inhibition of the HPA axis. Number two, research in both mammals and birds has shown that electrical stimulation of the hippocampus decreases plasma corticosteroid levels. For example, I would hear this study in which they investigated the effects of electrical stimulation at different levels in the rostrocaudal axis of the hippocampal formation of pigeons. So they measured the cord titers as an indicator of HPA axis activity. And as you can see here, the results showed that the stimulation markedly reduced corticosterone levels at different locations of the hippocampus. Number three, we know that damage to the hippocampus by lesions or by relatively complete hippocampectomy increases plasma levels of corticosteroids, delaying the termination of the stress response. For example, here, the same authors observed a change in circadian corticosterone rhythms as a result of lesions placed in both rostral and caudal locations in the hippocampal formation of pigeons. And we see here in white And they compared the effect of these lesions to the normal circadian corticosteroid rhythm in the same birds before these lesions. So we see that, the hippocampal lesions actually change corticosteroid levels. So finally, the avian hippocampus has been shown to have a high degree of plasticity. Studies show that chronic stress can impact the hippocampus, leading to changes in its cellular and cellular structure and the function. And this last point has been one of the main areas of investigation in our laboratory. So findings from this lab have shown that the hippocampus can serve as a biomarker for cumulative affective experiences, meaning that by using measures of hippocampal neurogenesis, we may be able to gain insight into the impact of cumulative emotional experiences in brain function. For example, research in both mammals and birds has shown that exposure to unpredictable chronic mild stress results in a significant reduction in the number of new neurons incorporated in the hippocampus, specifically, as we see here, in the caudal pole. And as I mentioned before, given the existence of this functional specialization in the mammalian hippocampus, we think that the avian caudal pole may be in control of stress regulation in birds, while the rostral pole may regulate the other functions. However, other studies from the lab investigating the link between keelbone damage and hippocampal neurogenesis in laying hens found that hens with keelbone damage had a significant reduction in neurogenesis, as evidenced by reduced double coating staining across both rostral and caudal hippocampus. However, important to mention here is that those hens that acquired fractures earlier in their lives, so those who had been living with a fractured keelbone for the longest time, had fewer double coating neurons, and this was more prominent in the caudal hippocampal formation. So now, my role in the lab for the past almost 4 years as part of the Chicken Stress Project has been to shed light on the role of the hippocampus in the stress regulation of birds. Specifically, my project aims to identify which hippocampal subdivisions are involved in the negative feedback response that regulates the HPA axis, and how the hippocampal fibers communicate with the hypothalamic periventricular nucleus. So far, this can be summarized into three main projects. In the first project, we tried to identify the subdivision involved in the negative feedback regulation. For this, we conducted immunohistochemistry studies investigating the expression of CFOS, and we see it here in the images, which is a marker of neuronal activation across different regions of the avian hippocampus. So adult hens were restrained in a hanging bag for 30 minutes and returned to the flock for an additional 60 minutes. We collected brain for the immunohistochemistry, but we also collected pituitary for the analysis of stress-related genes of interest, and also blood samples. We took blood samples before the stressor, 30 minutes after stress induction, and 60 minutes after stress termination to quantify corticosteroid in plasma. So the results from this experiment show that the comparison between stressed and unstressed animals revealed that the rostral region of the hippocampus, as we see here, had a higher density of CFOS positive cells in response to the stress treatment. This response was similar across the different subdivisions studied, the ventral, the dorsomedial, and the dorsolateral. However, this was an unexpected result, since our prediction, as I mentioned, was that the ventral region would be activated in response to stress. After analyzing also the activation of other brain areas related to stress, as well as the genetic markers of stress we obtained from the pituitary, they actually didn't show significant differences between the groups. Additionally, the analysis of corticosterone levels showed that the animal's reaction to the stress treatment here was actually very variable between individuals. Leading us to suspect that the hippocampal activation we observe here is unlikely due to the stress treatment that we use, and maybe the result of other factors, for example, the differences in environmental cues during the stress induction. And here I'm gonna go back to the experimental setting for a minute because, I want to mention that before the experiment, our hands were located inside a pen in a room different to that in which we exposed them to the stressor. So this means that before stress exposure, we took them out of the pen, transported them through a hallway, and then restrained them in a bag in a different room. So if our hypothesis is correct, this means that the rostral pole of the hippocampus could have been responding to these novel environments, resulting in an increased C4 sustaining and surpassing maybe the effect of the stressor. But despite the results, our findings represented an important step towards understanding the complexity of the hippocampal function and highlighted the need for an improved experimental design in subsequent studies. So we did some changes, and I want to say that despite the challenges we faced in our initial attempts, we made significant progress, and we are getting closer to identifying the hippocampal subdivision involved in stress regulation. So for the second project, we took a different approach and decided to analyze the gene expression from the hippocampal tissue microdissections obtained from broiler chicks exposed to conspecific isolation. As the experiment before this one, the idea was very similar. So we took hippocampal samples at different levels within the rostrocaudal axis and from different subdivisions of the hippocampus. So basically, we took what we learned from the first experiment and included several control groups with identical environments to our test animals in order to reduce possible interference of environmental cues. We also performed additional behavioral tests to account for individual personality deviations that could potentially impact our results. And in fact, just a few days ago, I finished the PCR analysis for this experiment. So unfortunately, I can't share the results just yet, but so far it all looks very promising. And as a last project, I would like to mention some of the exciting results we have so far from the third project. This project was in collaboration with Loreta Medina, Esther Desfilis, and Alessandra Pros from IRB Lleida, where we performed ex vivo track tracing studies using organotypic slice cultures of chicken embryos. So for this, we targeted different regions of interest in the avian hippocampus and other brain regions involved in the stress response. So biocytin crystals, seen here, were placed in the PVN, while Texas red dextranaminas were placed at different subdivisions of the hippocampus. Overall, we obtained a highly consistent labeling pattern, and I will briefly show you one of the most representative cases. So our results showed retrograde staining of cells in the medial and central extended amygdala, the septum, and the ventral hippocampus. Here you can see the injection site, and we see that in the extended amygdala, labeled cells were located in the lateral and medial parts of the bed nucleus stria terminalis. These findings are consistent with pathways previously identified in mammals and birds, suggesting the communication of the ventral subiculum to the PVN via possibly the bed nucleus of the stria terminalis. While in the septum, labeled cells included mostly the medial and submedial septal nuclei, as shown here. And what is truly remarkable and exciting about our findings is that although previous studies in pigeons had already revealed indirect projections from the ventral hippocampus to the PVN, our results suggest that a direct connection may also exist, and this expands our understanding of the neural circuitry underlying stress response in birds. We see here very clearly marked the ventral hippocampus in the chicks. So just as a conclusion, future research may shed more light in the involvement of the avian hippocampus in the stress response, and comparative neuroanatomical studies will certainly be a key on covering these structural and functional differences. And understanding the evolution and neuroanatomy of the hippocampus in different species can provide important insights into the underlying mechanisms of these processes, as well as potential avenues for treating disorders that involve hippocampal dysfunction, and maybe novel insights into stress resilience. So with this, I conclude, and thank you very much. I will be happy to answer any of the questions you may have. Thanks.
All right. Thank you very much, Karina. Does anybody have any questions for Karina?
Hello, Tom. Can I
Please
ask a question? Yes. Yeah. Okay. Hello, Karina. Thank you for this beautiful talk. And my question is just on the nucleus of the hippocampal commissure, where you also have cells. What do you think about that nucleus?
Yes, yeah, I was just gonna show one of the cases, the 20C1 that you're familiar with. And I went back and yeah, I didn't see a lot of cells there. I may have the wrong image, but I honestly put it together in the last minute. So I may need more time to go do a close-up into that brain. But yeah, I think, yeah, I remember we saw some cells in the nucleus of the hippocampal commissure, and this is also very exciting because we know that it has also been proposed to participate in the stress regulation. So this could actually give us more insight into its function.
any other questions for Karina? Daniel, hold on, let me make sure that you can actually unmute yourself. Go right ahead.
Hi, thanks for that really interesting talk. I was just wondering if there's any work, or if you're planning on any work on different chicken breeds or varieties. I know there are some varieties that, you know, can't stand up properly because their breasts are so overgrown and this kind of thing. And to me, that would naturally be very stressful. And so I was wondering if there's any interest in sort of the ethics of that and the stress of that.
Yeah, I understand what you say. Yes, well, our first experiment was with laying hens, and the second experiment was with broiler chicks. So these are the broiler ones are the ones that grow very fast, are very muscular, and yeah, compared to the laying hens that have almost no muscle and they are just production. And yes, there are also differences between more ancient kinds like red jungle fowl compared to white hens. But yes, we are not exploring particular breed differences in these studies. We want basically to know like the anatomy of the hippocampal formation, but we're not going very deep into these differences between breeds.
Can I just say that would be a really interesting thing to do?
Yeah.
So we know, for example, that white laying hens and brown laying hens are very different in their behavior, in their response to stress, in their response to all kinds of environmental stimuli. And so even there is a very interesting comparison, but we need the basic anatomy before we start looking at quantitative differences in how these things actually connect to each other and control each other.
I'm very, I agree totally with what you both said. I would be super interested in differences in brain anatomy, not just brain function between different breeds. Sort of the idea that if you're selecting for different body types and body structures, you can accidentally get different, select for different brain anatomies along the way, like you mentioned, different behaviors in the brown and the white-laying hens. I'm also very interested because of the comparative aspect in getting the ancestral bird, the wild type, the wild red, what's it called?
Jungle
Red
fowl.
Jungle fowl, yeah.
Yeah, and looking at their anatomy and comparing it to all these different breeds, I think there's a very interesting parallel, very interesting case there, which I think they're doing in dogs, but not in birds.
There's a little bit of work being done by Per Jensen in Sweden, comparing red jungle fowl to more domesticated breeds in gene expression and some neuroanatomy, but not very detailed neuroanatomy at the moment still.
Yeah.
So might be worth looking at his papers. Any other questions? Seema. All right, Seema, let me make sure you can ask a question. There we go.
Hi, thank you for that nice talk, Karina. I was interested in the corticosterone response that you said, the variability in the corticosterone response to the stressor. If I'm remembering the graph correctly, it does seem that the recovery points were lower than the baseline points, which supports your thinking that some of the animals might have been stressed before they started. I mean, there's all, maybe looking at other hormones would be useful, ACTH or something like that, or maybe an additional time point. But is there a possibility that there are like really strong variations between chickens? I don't know enough about them.
That is a very good, that's a very good question. And yes, one thing that surprised me of working with chickens was to notice that they have actual personalities, and I was not expecting that. That was a surprise. So from this first experiment, that was one of our main outcomes. So in the second experiment, what we did before the stressor, actually we did a personality test. So I classified my animals in that experiment into high responders, low responders. I did like a gradient according to the calls that they made when I separated them. And yes, so we have the videos, we have the calls, and we have these personality tests. And the second experiment may give us more insight into if these high and low responders actually react different to our stressor. Yes, so very exciting.
Great, yeah, very, very interesting. Look forward to that. Thank you.
Thank you.
Right, Spock, last question.
Last question. Karina, really nice job. Thank you for doing this. I was really excited to see you use the keel as I guess a non-neuro kind of measure to put there to kind of look at what differences there may be. And I'm wondering whether you perhaps you're thinking of this, or perhaps you have this data already, if there was any differences in the laying down of bone that might correlate with this. For instance, like in humans, there's been a lot of work in primates on the laying down of enamel and long periods of chronic stress and how this tends to impact another organ system. I can imagine that if we're devoting a lot of time towards one system versus the other, that there might be some parallels that you might see there. I don't know, Tom.
Yeah, I think Tom may have a better response because I'm leaving the lab in a month, so I don't have future plans.
That one was definitely the other way around. So the keel bone damage is a very common thing that occurs in commercially housed chickens, especially in the new quote unquote cage-free environments where there's more opportunities for them to basically fall off a perch or hurt themselves. Laying hens constantly live on a very knife edge in terms of calcium balance because they need to make eggshells every day and they need to keep their bones healthy. That does mean that in principle, that balance could be very easily pushed over by chronic stress or not, but we've never thought about using, kind of layers in the bone as a record of potential stressors in an animal's life. Now, interestingly, we have all those keel bones sitting around of those birds, but of course that was to quantify the damage that was done to them and how many times they broke their keel bones rather than anything else. So it's intriguing. I will talk to my more poultry-like friends and see if they know anything more about that, people who know more about the other bits of the bird than just the brain. Thank you, though. All right, this is probably a good point to take our break. Here in the UK, it's 10 to 6. Wherever you are, it's 10 to something. It's dinner time for us. I'm sure it's lunchtime for the people on the East Coast of the US. Everybody else, do what you like with your particular food. We will reconvene in about an hour and 45 minutes at what is here 7:35. For you, that might be some other 35. Let me just make 100% sure I'm not saying anything wrong. Yes, at which point Sina Bhatnagar will give her talk and then Carrie Fegdor will follow from there. I will be back a good 10 minutes, if not more, before that, Sima and Carrie, if you want to try out your slides and all that. So I'm going to stop the recording now. I'm going to put it on Spock to remind me to restart the recording when we come back. I'll try, of course, as well, but it's always good to have an extra person to help me remember. So I'll stop the recording and